Dear colleagues, my name is Anatoly Sorokin, and I would like to present here the new way for analysis of functional characteristics of microbial communities with flux balance partial random correlation coefficient. Microbial communities are omnipresent. They play a important role in ecology, in medicine, via the interaction between human body and its symbionts. And recently it acquired a lot of attention from a biological community, especially for the participation in microbial fuel cells and bioelectrical systems. The key obstacle in wide implementation of biological communities in biotechnology in particular is that we are vaguely understand the governing principles which define the structure of the community and its kind of controls its stability. One theory which describes the structure of the community is called community coexistence theory, which is assumed that community has stable structure. That structure is defined by interaction between species and interactions mediated via metabolite exchange. And that exchange could describe the competition when more than one species consume the same compound. Mutualism when there are obligatory cross-fitting between two species, and commensalism when one organism depends on the other for supply of the nutrients. In terms of modeling, community coexistence could be, there are two major approaches. One is called reverse ecology, which is graph analysis of whole genome reconstruction of metabolic network. And another one is community flux balance analysis, which also use whole genome reconstruction of metabolic network, but in the form of constraint-based modeling. For community FBA, models of individual species combined via shuttle reactions which describe exchange of the metabolites from species to the environment. And that exchange is controlled by two values: lower boundary of the reaction limit how much metabolite could be consumed from the environment, and upper bound describes how much it could be sent to the environment. We use that two values to model the interaction between species within community, and use sensitivity analysis, which determines how different values of independent variables, in our case lower and now upper bounds of the exchange reactions, influence particular dependent variable, in our case biomass production of particular species. We use partial random correlation coefficient as sensitivity measure, as it provides both sign and amplitude of the effect and could be effectively calculated, and its significance could be easily assessed. Recently, we published application of global sensitivity analysis to flux balance analysis for optimization of final product for biological E. coli. And as I mentioned, from the sensitivity analysis, we have two type of variables, upper bound and lower bound, and we got two signs of the coefficients. Usually in coefficient analysis, people consider only consumption productions, which corresponds to the positive correlation coefficient between in and out flux of the compound and biomass production. But we also have examples when the correlation coefficient is negative, and in this case that could be interpreted as attempt of the cell to minimize influx or outflux of particular compound, which could correspond to looking for areas with lower or high concentration of particular compound. And because of this, we call that two interactions attraction and avoidance. And for example, if we when we analyze the microaerophils, we kind of which tend to avoid high concentration of oxygen, we kind of the lower bound uh, Clausius coefficients for oxygen is negative for that species. Uh, to capitalize on compositionality of our approach, we calculate the big graph of species metabolic interaction uh, based upon collection of Agora semi-manual reconstruction of whole genome constraint-based models. Uh, we use 800 bacteria, and at the level of significance of 10%, we have 800 bacteria and 700 exometabolites, 35 of which are inorganic. The central part of the graph looks like presented on that slide. Different colors represent different type of nodes in the graph, and size of the node correspond to importance of the node based upon betweenness centrality. Analysis of the uh, different type of edges in the graph shows that 80% of uh, interactions within the graph correspond to consumption, which is expected because the main source of information for reconstruction of whole genome flux balance model is the information about ability of bacteria to consume particular source of carbon, nitrogen, and so on. What's interesting that the level of attraction and avoidance is almost the same as level of production. Because we have just introduced that concept, we expect to be number of that type of interactions much lower. To conclude, we propose new approach to model interactions between bacteria and metabolites within the community based upon global sensitivity analysis of flux balance models. Uh, we uh, build a large graph of such interactions based upon collection of 800 human microbiome related species, and we introduce two new species metabolite interaction modes, attraction and avoidance. Thank you.